Thank you to everyone for coming. My name's James Chong. I'm a cardiologist at Westmead Hospital, and I'm a research scientist here at Westmead Institute. And tonight's event is one of our Meet the Researcher events, and it's a really great initiative by the Westmead Institute where we get to talk to members of the public about the research that we do and explain why we do what we do. So just to give an idea of the order of proceedings, I co-direct the Centre for Heart Research with Associate Professor Eddy Kizana. I'll be giving a short talk about the Centre for the Cardiac Regenerative Laboratory, which I lead as part of the Centre for Heart Research. And then one of my staff members, Dr. Zoe Clayton, will give a short talk. Eddy Kizana will then give a talk about framing his side of the group and why he does his research, followed by an ex-PhD student of his, Melad Farraha, who will be talking about his research in a little bit more detail. Then, at the end of the night, we will have a really important part, which is a question and answer session, where you get to ask any questions that you might have of us about our research. So, without further ado, I'll hit into... So, I've introduced who I am, and as mentioned, I'm a cardiologist. I'm an interventional cardiologist, and that really drives the research that I do. So, for this short presentation, I'll be using an actual case to try and explain why I do the research that I do. And this case is a gentleman who came through the hospital. It's about 4 odd years ago now. We're going to call him Joe for this situation. And he's in his late 50s. He came in with crushing chest pain. And he didn't realise it at the time, but he was having a heart attack. And heart attacks are caused by a lack of blood flow going to part of the heart. And as an interventional cardiologist, so we come in any time in the day or night and perform a procedure known as a coronary angiogram. And that's what you're seeing here up on screen. You can see the pointer. These are the blood vessels to the heart. We use X-ray guidance to see the blood vessels. And what you're not seeing here is a blood vessel that should come down the main, the front of Joe's heart, and that's because it's blocked. That causes the heart attack, but fortunately, we become pretty good at treating these problems. So again, as an interventional cardiologist, I'll go in and put a little wire into this artery and blow up a little balloon in the artery and to restore flow, and then leave a metal sleeve called a stent to hold that open. And now you're able to see that there's blood flow restored to this artery. Now, Joe didn't actually realise he was having a heart attack, and that's not uncommon. Many of our patients say, oh, it wasn't pain, it was indigestion, or I didn't know what was going on. And Joe, because of that, Joe was actually having heart muscle damage, and part of his heart was dying before he even came to hospital, and before we were able to open up flow. And that's a problem. This is an ultrasound of Joe's heart the day after the procedure. Now, I'll just orientate you. This is the main blood pumping chamber of the heart, and what you're not seeing is contraction, beating of the main blood pumping chamber all around here. Really, that should be like this here, pumping vigorously. And this creates a situation known as heart failure. One of the heart's main roles is to pump blood around the body. And if your heart's not doing that, if it's failing, you get very sick. And that's what happened to Joe. So because he was sick, he ended up in intensive care with that very poorly functioning heart. And for many patients like him, the only option for severe heart failure is curative treatment for severe heart failure is a heart transplant. And many of you will know that although that's a very good treatment, there's just not enough donor organs to go around. And for Joe in particular, he had some other issues with his lungs, etc., that didn't even make him a candidate for a heart transplant. And that's the situation for many of our patients, especially as we get older. There's not enough donor hearts to go around. So unfortunately, Joe died 3 days after we restored flow into the arteries to his heart. There are many patients like Joe, though, who actually do end up leaving hospital, but they leave hospital with that poorly functioning heart, and they live with this condition known as chronic heart failure. And life's very bad if you have chronic heart failure, with severe heart failure. Blood backlogs into the lungs, and that gives a sensation of almost drowning, shortness of breath. They have difficulty getting out of bed and doing the things that you and I take for granted. So that's a major problem, and that's part of why we do the research that we do. Here's some statistics on heart attack in particular. As you can see, it's a major killer of Australians and people around the world. It peaks in mid-age. Many people think that heart attack's a problem for men, but as you can see from this graph, this is Australian statistics, recent Australian statistics. You can see that women are almost equally as affected, but potentially it's just slightly delayed. And the condition of heart failure that I mentioned, here's a statistic that might shock you. 1 in 2 patients with severe heart failure, that's once it gets to the level that, like Joe's heart, die within 1 year of diagnosis. And that's a category that places it in a poor prognostic group, worse than many cancers. People, I don't think, quite realise that about heart disease. Sorry, technical issue. This was the 1 in 2 patients with no. Okay, this is it. Sorry, 1 in 2 patients die within a year of diagnosis. And why do we have this problem? Well, it's partly because the heart, together with the brain, is one of the least regenerative organs in the body. And when I was in university, at the University of Sydney, and when I was a junior doctor at Westmead Hospital here, I was taught that the heart couldn't repair itself, couldn't regenerate itself. And then it's only later that we've actually realised that that's not exactly true. That's where this little guy comes in. I'm not sure if anyone recognises this. This is an axolotl, also known as a Mexican walking fish. Now, why I brought him into the story is because these amphibians have a remarkable ability to regenerate organs. They've found that they're feisty creatures. They actually attack each other, bite each other, and if they lose a limb, they can actually regrow part of that limb. And researchers have actually worked out that you can cut off part of their heart and they'll regrow part of their heart. We've also found out that mammals have this capacity as well, mice and humans as well. Now that's important because it sets a precedent in biology that we're trying to harness in the Westmead Institute. So we're trying to regenerate the heart after injury, and part of the way that we do that is using stem cells. Many of you might have heard about stem cells but not known exactly what a stem cell is. So in its simplest form, a stem cell is a cell that's able to reproduce itself, but it's also able to produce other types of cells. And you can see from this cartoon that we can then use that in our research to produce cells that are important to cure diseases like heart failure. So in particular, we're trying to build heart muscle cells. They're known as cardiomyocytes. Now this is a video of what you might be seeing as though you were looking down the microscope up on level 6 in our laboratory upstairs. This is beating heart muscle. It's insidia. They beat in the dish. This dish is about so big, and we can create maybe 10 million heart muscle cells from stem cells just in this dish. And we're literally producing these by the billions in larger flasks up in the lab by many of the group who are sitting down the back there. So you could see that because the stem cells are able to replenish itself, If we look after that correctly, we can then produce an inexhaustible supply of heart muscle cells that we could then use to treat patients. And that's in contrast to this limited donor supply of hearts. So possibly, and it's an if, but possibly we could replace heart transplantation as a cure for heart failure. And that's a goal that we're trying to achieve. So bringing it now back to our patient that we started off with, Joe. This is an animation showing what might be possible. So heart attacks are caused by lack of blood flow going to the part of the heart. That lack of blood flow causes heart muscle death. And then, as I've shown you, in the lab we can then grow billions of heart muscle cells that we can potentially come back and transplant, inject into the patient to restore their heart function. Obviously, I've pictured the best case scenario, but it's exciting. There are clinical trials starting around the world, and we're aiming towards starting clinical trials for our own in Australia that will benefit our patients, the patients that I'm treating across the road. We're not there yet, but you can see that I'm optimistic, and this is what I'm trying to do. We're trying to give a new start, give a new lease of life for patients like Joe and those with chronic heart failure. So hopefully that's encapsulated a bit about what we do, and it's now my great pleasure to introduce one of the talented postdoctoral researchers in my group, Dr. Zoe Clayton, and she's going to go through in a little more detail some of the work we do.
Okay, thank you very much, James, and I'd just like to quickly thank the organisers of this Meet the Researcher event for giving me the opportunity to speak this evening. So I'm a postdoctoral scientist in James's lab, and I have a background in cell biology, specifically stem cell biology, and so I'm one of the people that grows these stem cells into heart muscle cells in the lab upstairs. And so what I'm going to talk to you about this evening is some of the details that we still need to work out before these stem cell therapies can be translated into the clinic. And then right at the end, I'm just going to tell you a little bit about another very promising therapy for heart failure, a growth factor therapy that our group has been developing here at Westmead as well. So we've actually known about stem cells for a number of decades now. Back in the early 1980s, mouse stem cells were first isolated from mouse embryos, and this was a major breakthrough, and it ended up winning the Nobel Prize. And then there was a bit of a gap for a few years before 1998, where we first isolated human stem cells from embryos. And then after that, the stem cell field really took off. And although this was a major advancement, it was also quite a controversial development because we were relying on human embryos as a source of these stem cells. So in 2006, there was another major breakthrough when we discovered that we can actually generate stem cells from normal adult cells, for example, your skin cells or your blood cells. And these are called induced pluripotent stem cells. And that breakthrough also won a Nobel Prize in 2012. And in the past decade or so, there's been a lot of first-in-man clinical trials with different types of stem cells for different disease applications, for example, spinal cord injuries, vision problems, and then in the background, people like James were making major breakthroughs, finding that stem cell-derived heart muscle cells could actually regenerate the heart, and that this results in a functional benefit to the heart. And in 2019, we're just beginning to start the first clinical trials specifically for heart failure patients, and this is going on in Japan and in the United States, and then hopefully soon in Australia. So, as James mentioned before, the heart is one of the least self-regenerative organs in the body. And we have about 2 or 3 billion heart muscle cells in an adult human heart, and that number stays relatively stable throughout your life. But during a heart attack, depending on the severity of the heart attack, hundreds of millions of these heart muscle cells are deprived of blood flow, so they're deprived of oxygen and nutrients, and they die. And so what we really need is stem cells that we can turn into these heart muscle cells, or cardiomyocytes as we call them. And what we really need to do to get them into patients is overcome a couple of issues. And these two issues are, one, poor cell engraftment. So what we know is that when we transplant cells, the majority of them do not survive, and they don't attach themselves properly to the heart muscle. And not only is this a limitation from a potential therapeutic angle, but also when we're trying to do clinical trials and commercialize these therapies, it's a major waste of resources if 90% of our therapy just isn't working. So we really need a way to improve engraftment. The second issue is arrhythmias. Eddy and Melad will talk to you a lot more about arrhythmias soon, but an arrhythmia, what you need to know is that it's a disturbance to the normal heart rhythm, the natural rhythm of the heart. I'm sure most of you recognize the normal ECG trace. And because the stem cell-derived heart muscle cells, they can actually beat independently, but if they're not beating in sync with the heart itself, that can interfere with the heart's natural rhythm and potentially cause arrhythmias. And the preliminary data we have suggests that these arrhythmias might just be temporary and they don't seem to be life-threatening, but when we're talking about using these cells in the clinic, we really need to minimize the risk of arrhythmias. We can't be too careful. And so what I'm going to do now is just take you through how we grow these cells in the lab. So we have our stem cells here. These are some cells that I've been growing upstairs. And we treat them with small molecules or drugs that encourage them to become heart tissue. So after about a day, they become what's known as mesoderm, which is the type of cell that all cardiac cells eventually come from. And we treat them with more specific drugs at various time points. And then by about day 5 or so, we start to see these clusters of cardiac precursor cells or progenitors. And then by day 7 to 10, we have a nice beating sheet of heart muscle that we can then use for our experiments. And so these are just 2 images of cells that we are using, cell lines that we are using in our lab. These were taken on a special type of microscope called a confocal microscope. And what's special about these cell lines is that they have fluorescent reporters attached to them. And what that allows us to do is track the cells. So when we transplant the cells, we can identify where they are. And in this line, this is our NKX line. NKX is a heart-specific gene. And it's got a green fluorescent protein attached to it. So we know that when our cells are fluorescent green, that means they're heart muscle cells. And we have here is our TD tomato cell line. TD tomato, as you can probably guess, is a red fluorescent reporter, and so we can use that to track our cells. And what we've also done here is stain them with a specific heart muscle marker called cardiac troponin T. And cardiac troponin T is part of the contractile machinery that makes the heart cells beat. And so because we can see that we've got this distinctive cardiac troponin staining, we know for sure that we have cardiomyocytes. But what might not be so clear from that video and from these images is that when we grow these cells in the lab, we don't actually end up with a homogeneous bunch of one cell type. We actually end up with a number of different cell types. And so what we need to do is kind of find a way to separate them out. Because as I'm sure you can appreciate, there are many different cell types in the heart. And as you may know, the heart is comprised of 4 chambers. We have the 2 major pumping chambers, the ventricles, and so we have ventricular-specific heart muscle cells. We also have cells more specific to the atria, and small populations of specialized cells called pacemaker cells. In addition to that, we also have cells that form blood vessels, cells that form smooth muscle, and a special type of supporting cell called a fibroblast. And fibroblasts form a sort of cushion for the heart muscle cells. They're also important in scar formation after a heart attack. And so when we grow these cells in the lab, what actually happens is we end up with a mixed population of all of these cell types. And as I'm sure you can appreciate, if we want to use a cell for a specific therapeutic purpose, for instance, if we want to repair damage to the ventricle, we are probably going to have much more success with a pure population of ventricular cells rather than this mixed combination. So what we've done is we've developed a new method to identify these different cell types and to separate them out so we can use them for downstream applications and we can use them potentially in heart regeneration. And the way we do this is by taking advantage of the fact that each different cell type has its own unique signature, in that each cell type expresses different proteins on its surface. And what we can do is we can bind other proteins called antibodies to the cell surface. And if we also tag those antibodies with a fluorescent label, then we can see what's being expressed on each different cell. And then we use a technique known as flow cytometry. And what this does is it takes all of the cells and it passes them through a very narrow tube, so they almost move through in single file. And then a laser beam shines through that tube, and as the cells pass through, the fluorescence gets detected. And what you end up with is an output that looks like this. And what's cool about this is that you can look at many different colors or cell markers at once, and each little dot on the plot corresponds to a single cell. But when it becomes difficult is when you have many, many colors at once, the data gets very complex and it gets very difficult to interpret. So what we've done is we've got a computer to do the data analysis for us. And so on the right-hand side here, we have our marker panel that we're using in the lab. We have 13 different colors, including the very aptly named zombie stain, which filters out our dead cells for us. And on the left-hand side, we have the computer-generated outputs. And what the computer has done is it's analyzed every marker expression on every cell, and then it's clustered the cells so that cells that are very similar to each other are arranged closely together, and cells that are not so similar are arranged further apart. And so we can identify different cell types based on where they cluster. And what the color gradient shows us is how much of each specific cell marker each specific cell is expressing. And we're pretty excited about this because this hasn't been done before in the heart regeneration field. But furthermore, we've also developed a new way to sort the cells based on these clusters, and that's been going on in conjunction with the flow cytometry experts here at WIMR. And so we now have a novel method that we're pretty excited about because it has a lot of downstream applications. And some of these applications that we're already working on is identification of the best engrafting cell types, to potentially use them as an optimized cell therapy for heart failure. We also see a lot of potential in this method for disease modeling and for drug testing applications across a wide range of cardiac diseases. And so now, just in the last couple of minutes, I'm going to switch topics entirely, and I'm going to tell you about another highly promising therapy that could potentially be used in conjunction with cell types or even as a standalone therapy for patients who've had a heart attack, and that's platelet-derived growth factor therapy. So platelet-derived growth factor is it's a protein. It could be a drug that binds to a receptor on the outside of the cell, and when it does that, it changes the gene expression of that cell so that that cell starts to divide, it migrates, it makes new blood vessels form, and it increases the deposition of things like collagen, so the structural materials that all cells and tissues need to actually grow and function properly. And so what Sujita, one of the PhD students in the lab, discovered was in a preclinical study, if you treat the heart with PDGF, it actually improves the heart function and it improves survival after a heart attack. So these are just MRI images looking at the left ventricle of the heart after a heart attack. And I don't know if you can see here, but is it working? Anyway, it's contracting very sluggishly, and there's not much movement of the wall as the heart contracts. But when they were treated with PDGF, that movement became much more forceful. The function of the pump function of the heart was restored. And we think that this is happening through the growth of new blood vessels, and we think that PDGF is also acting on the scar and essentially making a better scar. So using a number of quite cool microscopy techniques, we've shown that the PDGF treatment increases the number of blood vessels in the damaged area of the heart, and we can see that in 3D here. This is 3D image rendering showing the blood vessels in sections of the heart, and it also makes the scar line up a lot better, which makes the scar better for the heart, functionally for the heart. And so what we think is going on is that PDGF is acting on some of these cell types, the fibroblasts and the blood vessel cells, and it's altering the structure and the composition of the scar, and it's promoting new blood vessel growth. And what the outcome of that is improved heart function, reduced arrhythmias after a heart attack, and ultimately reduced risk of death following a heart attack. So I just want to finish up by saying thank you to all of these people. This is, between the two pictures, I think this is just about everyone in the Centre for Heart Research, and I also just want to particularly thank the Westmead flow cytometry team and our collaborators who help us out with the cells. Thank you. So I think we're going straight up into our next speaker, who is Associate Professor Eddy Kizana. He heads the gene therapy group, and he's going to talk to you about cardiac arrhythmias.
Thank you, Zoe. So I'm going to talk to you about the work that I do here in the Institute. And really, like James, I come from a clinical background as well. So I work next door at the hospital as well. And my interest in cardiology is around the electrical function of the heart. And a few years ago, the last talk I gave here, Kevin Sheedy was in the audience. And he came up to me at the end of the talk and he said, "You're the smartest electrician that I know." And essentially what I do is fix electrical problems in the heart, so diagnose and treat them. So the purpose of my research really is to use a technology related to stem cells, but called gene therapy. So we go smaller than cells, we go below down into the genetic structure. And we use genes to treat heart rhythm problems. And in fact, what we do is try and rebuild or regenerate the electrical conducting system. And the lofty goal for our research here is to try and replace electronic devices like pacemakers and defibrillators, which is a far cry from the type of therapy that psychology envisioned for us. Sigmund Freud pictured there talking to a gene. Like James, I'm going to talk about a couple of patients that really highlight the limitations of current technology. This is a patient like Joe, but patient XY, who's a 56-year-old man who had a heart attack, and we can say James was on call that night and came in and treated Mr. XY and promptly opened up his artery, and you can see there's blood flow restored down the front wall of the artery. However, unfortunately, like Joe, he presented fairly late and developed scarring in the front wall of his heart. Yep, there it is there. It's showing that the contractile function of the heart is reduced following the heart attack. And being an electrician, we were interested in the electrical function of the heart following this heart attack. And what this image or video shows is that there's a big scar on Mr. XY's heart. The purple area is showing normal muscle, whereas the red is scarred muscle, and the colours in between, need to go back. Let's see if I get this to play again. There we go. And those sparkles in between are showing the flow of electricity. And what this type of flow is showing is an abnormal heart rhythm called ventricular tachycardia. And the reason why ventricular tachycardia is very important is because this is the reason why people drop dead after they've had a heart attack. And so once we found that Mr. XY had this problem, he was treated with conventional treatment. And at the moment that consists of a defibrillator, which is implanted under the collarbone with leads that go down into the heart, an array of different medications to strengthen the heart and to prevent the electrical disturbance from happening, and also catheter ablation, which some of my colleagues in the audience here perform on these sorts of patients. And it works, but it doesn't work in all patients. And some patients are left with residual scarring, with residual electrical abnormalities. And unfortunately, if they don't get transplanted, they can succumb to those sorts of problems. And this is despite having defibrillators inserted. So this is the first patient. The second patient is Miss XX. And she's a 35-year-old woman who presented to our hospital with collapse. Unfortunately, an ECG was recorded at the time that she collapsed. And what this shows is only a few electrical beats. So during the episode, her heart stopped completely. There was electrical standstill. And this is the cause for her collapse. And again, she's at risk of dropping dead and dying suddenly. She had some inpatient investigations done, and some of the people in the audience might recognise these images. She had an MRI which showed an abnormal signal on the back wall of her heart. And this is an important part of the heart, and I'll show you why in a second. But she had a workup done, and she was diagnosed with an inflammatory condition that can affect the heart. And again, she had an electrical study done. And again, pink is normal, red is scarred. And unfortunately for this patient, the inflammatory condition had damaged the part of the heart where electrical signals originate. And this is the sinoatrial node. So this is where electrical signals start in the heart, and this is where she had her disease, and which meant that her endogenous, or the inbuilt pacemaker system, was destroyed. So again, she was treated with a pacemaker, an electronic device. She was given medications to limit the inflammation from the disease and spread it and stop it from spreading to other parts of the heart. But the questions arise: how long does a pacemaker battery last for? Depending on how often it's used, it will last between 5 and 10 years. So patients who use it a lot, it'll last around the 5-year time point. If you use it a little, it'll extend the life for about 10 years. So imagine someone at the age of 35 having a pacemaker implanted that lasts for 5 years, and she's expected to live to the age of 80. That's several devices over her lifetime. And if you're a child who needs one of these devices, and there are children who have them implanted, then the revision surgeries are great. So I've highlighted some of the limitations of existing technology. I don't want to put them down too much, but they do work, they do save lives, but there is a limit to what they can do. And what we're trying to do here in this institute is go beyond that limit using technologies like gene therapy and stem cell therapy. And my group is really focused on using gene therapy, as I said, to regenerate the electrical system. The strengths of gene therapy is that it can target the disease at the underlying genetic level. The genes are packaged in nano-sized particles, which means that they can be injected into the bloodstream, injected directly into the muscle, and most importantly, that these gene therapy particles can cause long-term effects. And by long-term, I mean over several years. So conceivably, instead of someone having a pacemaker implanted, they could have a gene injection performed, which turns their heart muscle into a pacemaker, and that's the end of it. No electronic hardware whatsoever. Just by way of acknowledgement, so I don't work alone. I've got a picture there of the group, and on the left is a number of staff members from the clinical department as well. So this is really just to emphasise the translational nature of the work that we do here at the Institute. And after me, I think Melad is presenting next. Melad, by way of introduction, was a PhD student. He's worked from the ground up developing the biological pacemaker system. He got his PhD earlier this year. Congratulations, Melad. He is at the moment a medical student at University of Notre Dame Med 1, but we've kept him on board here to continue to develop this project, which he's very passionate about. And I'd like to invite Melad up to the podium to tell us a bit more about his work.
Okay, thanks Eddy for the introduction and for the organisers for actually allowing me to give this talk today, just to present some of the work that I undertook as part of my PhD, as well as some other work that our lab is actually working on. So as Eddy mentioned, my name is Melad, and today I'm just going to be talking about advancing the development of gene therapy for cardiac arrhythmias, but more specifically on my project where I was looking at developing a biological pacemaker. So to begin, the sinoatrial node, as Eddy mentioned, is a group of specialised cells that have the ability to keep our hearts beating regularly. This is also known as the pacemaker. This pacemaker, as Eddy also mentioned, can become defective by means including heart attacks, heart disease, genetic defects, but most prevalently because of ageing. To date, there are no known cures for pacemaker dysfunction, with the only viable management option being the insertion of an electronic pacemaker. As Eddy also mentioned, there are issues with inserting electronic pacemakers, and these include subsequent surgeries required to replace faulty pacemakers or leads. These are electronic devices and as such require their batteries to be replaced every few years. There is lack of biological control because the device doesn't actually integrate with the body, but most seriously, infections can take hold. And as such, a more refined treatment option is required to manage pacemaker dysfunction, and to also reduce the risks associated with electronic pacemaker insertions. And this is where my work fits into the picture. So the aim of the project was to correct pacemaker dysfunction by developing a biological pacemaker using gene therapy. And to do this, we essentially borrowed from nature by packaging the pacemaker gene into a gene vector and then delivering this gene into normal heart cells to convert them into pacemaker cells. Now, these converted cells could then replace the dysfunctional cells in the heart to restore the regular heart beating. This is nicely depicted in the cartoon above, because we can see that the gene vector is delivered to the normal heart cells, which can then over time convert into the pacemaker cells. And to the right of that, we can also see what these normal heart cells look like under the microscope, and then how they look like once they convert into pacemaker cells. Now, as you may appreciate, this is quite a complex task to undertake, but it was broken down into 3 main sections to logically work through in these experiments. So the first step involved building the gene vector from scratch to contain the pacemaker gene and showing that I could produce it to a large enough amount that would be functional. The second step was then to add this gene vector to normal heart cells and plate them out in special tissue culture plates in the lab where the gene could then exert its effect on the cells. And then after 3 weeks, the third step involved analysing and imaging these cells under the microscope to confirm that they had converted to pacemaker cells and that they were functioning as we expected them to. And so over the next few slides, I'll just summarise the main findings that we got from this third step. So the first result depicted here is to confirm that the gene vector I created was actually functional and behaving as we expected. So what we have here is an image of some cells that have been stained with a special dye to locate the presence of the pacemaker gene. After which we added the gene vector to the cells and maintained them for 3 weeks. If the gene is present, the cells would stain pink, and if the gene is not present, then it would be more of a blue colour. And so from this image, we can see that about 90% of the cells had the pacemaker gene present. So satisfied that the gene vector was behaving as we expected, I proceeded to actually perform the experiments to cause the conversion of the pacemaker cells. Now, the most apparent distinction between heart cells and pacemaker cells is their size and shape. Above, I have an example of what my normal heart cells look like under the microscope when they were cultured in these special dishes in the lab. They were thick, large, block-like structures that take up a lot of space. I once again added the gene vector to these cells, expressing the pacemaker gene, and maintained them for 3 weeks. And then once the cells underwent conversion, they became like this. So they became thin, long, spindle-shaped cells, which are indicative of pacemaker cells. And we analysed the width and area of these cells to show that a very large change was occurring as we had hoped. This was very strong evidence that the cells were truly undergoing conversion and that our gene vector was performing its function. However, the results to this point were all well and good if the cells are converting. But the most important thing is to confirm that the cells are actually functioning like pacemaker cells. Now, if you recall back to the beginning, the pacemaker keeps the heart beating regularly using electrical impulses which travel throughout the heart. The way these impulses originate from the pacemaker have a distinct signature, which is known as the action potential. And from here we can see that, so using this complicated microscopy technique, we can see that analysing the electrical impulses from these normal heart cells, we have an example of what the impulse looks like. And so it's more of a square shape, there's quite a big gap between the rise and the fall, and this is indicative of normal heart cells. I once again added the gene vector and maintained them for 3 weeks. And after 3 weeks, there is a stark difference in the shape of these electrical impulses. And this confirms that the cells were changing on a functional level to behave like pacemaker cells once we had added the gene to the normal heart cells. And so again, this sort of proves that our cells are both functionally and morphologically and physiologically changing into pacemaker cells, which we can use for the next phases of our experiment. So to summarise this body of work, we can see that we were able to successfully construct a gene therapy vector for delivering the pacemaker gene to the heart cells. I demonstrated that we could convert the cells from the heart cells using our gene vector, and then that the cells were also behaving like pacemaker cells as we expected. Now, as I mentioned earlier, this isn't the only area that our group is working on with regards to gene therapy and correcting heart conduction disorders. Another area our group is working on is using or trying to re-establish the electrical conduction across the heart once the conduction channels are destroyed after an event like a heart attack, as Eddy mentioned. These result in very serious rate and rhythm issues and potentially have fatal complications if they are left unmanaged. So in this image, we can see what normal heart muscle looks like in a schematic with the connections between the hearts. This is where the cells allow for electrical conduction to occur across the whole heart. And these channels or conduction channels are known as connexins. Once the heart experiences a heart attack, the heart muscle dies and these connexins are broken down and lost. And loss of these conduction channels then result in slow conduction of the electrical impulses across the heart. This results in the life-threatening complications that Eddy had gone over. So our group is therefore trying to re-establish these conduction channels using the same principle as the biological pacemaker. So here, we have created another vector which contains the connexin gene rather than the pacemaker gene. And we have delivered this vector to the heart muscle cells, allowing it to re-establish these connexins between the cells and restoring the conduction across the heart. And then this allows the conduction to occur at a much faster rate, and also again preventing these life-threatening complications from occurring. And we have to date seen some excellent results with these experiments. So in terms of the long-term goals of these experiments, we hope to hopefully translate these into human patients, like the ones that Eddy had spoken about, that are afflicted by pacemaker and conduction issues. And so the procedure we plan to adopt already happens in patients, for example, when they undergo an angiogram or other cardiac procedures in the hospital. And it's a non-invasive method because it is done through the blood vessels in the groin, and it allows us to deliver the gene vector directly to the heart from the inside, rather than having to open up the chest wall and perform heart surgery. And so just to walk you through this video, the catheter is inserted into the blood vessels of the groin and advanced up into the heart. Once it's connected to the appropriate artery of the heart, the gene vector is injected into the heart and allowed to perform its function over a number of weeks. And this is schematically depicted quite simply, but it's much more complicated than that. And so on that note, I'd just like to acknowledge everyone that's assisted me with these projects, including Supervisors Eddy and James, fellow colleagues from the Gene Therapy Unit, from Centre for Heart Research, as well as the collaborators across the campus and the facilities here at the Westmead Institute. Okay, so on that note, then I'd like to invite Eddy, James and Zoe back up onto the stage. And we'll just have a Q&A now with anyone that has any questions. We also have the lovely Alison passing the microphone around. So if you have a question, just raise your hand and we can hopefully get around to everyone. I'll stay here. That's right.
So really, no questions too small or too big. This is really what this evening's about, where anyone can ask anything, and it's exciting for us to be able to share some of the work we do. So please.
James, I'll ask a question. You mentioned in your presentation that we're very close to human clinical trials for the stem cell research that you're conducting. How far off do you see that is, and what are the steps and things that are needed to get us there?
So I said no question is too small or too big, but Katrina's asked the question which I potentially dread. She's asked, how far are we from actually doing human studies? And why I dread that is because in science, things don't always work out. We have anticipations, we try and set ourselves mammoth tasks like the talks you've heard. The way I can answer that is that it's an exciting time in my area. There's trials starting around the world with support from people like the National Stem Cell Foundation and the G&K Foundation. We can make a dent into it. I think we are maybe For the stem cell type therapy, maybe 5 years for our own Australian studies. Now, as I've said, people around the world are starting their own trials now. So they might come sooner than that, but there's a lot of difficult problems. Zoe's mentioned the engraftment issues, that's getting the cells to stick, solving the electrical issues, Zoe's mentioned, but then there's all other less scientific problems that we have to solve. Trying to make sure it's safe, getting things through the regulators, that's known as the TGA in Australia. They're people who enforce that when you're going to treat a patient, it's not going to cause problems. Then it's trying to get the money to perform these trials, and that's very significant. So usually you're trying to generate investment potential and get investors to back it. And these are all hurdles which we're trying to attack. I hope that's answered.
Are there any problems with negative immune responses to these cell types?
Yeah, absolutely. That's a great question. So, yes, in short, we in our preclinical studies we use immunosuppression, which is what people do use for whole heart transplantation. Our hypothesis is that given that the load of foreign cells is lower, that we would need less immune suppression, but we think at the start human studies will need immune suppression, but it will be lower and it could be tailored off. That's a good question.
Do you
Great.
collaborate much with the groups overseas who are doing similar sort of work?
Yeah. We do collaborate with other groups, so I have strong networks with my previous mentor in Seattle in the United States. We held for the first time a National Institutes of Health meeting. First time it was held outside of the US. This was for heart regeneration in Parramatta in Western Sydney. And we had groups from Japan, Europe, Singapore, China, and our group collaborates with some of them, not all, but science, as you know, is a very collaborative process. We can only achieve things if we work together, and we do that internationally and nationally.
So when you put that research into the clinical practice, do you also observe the lifestyle of the patient, such as alcohol, smoking?
I can try and answer that. Yeah. So look, these therapies are premised, I think, on the basis that all patients have had contemporary medical therapies, including lifestyle changes, given to them or afforded to them as best as can be. So, medications have been given, devices have been given, lifestyle factors have been looked at, and I think the patients that we're talking about, all these things have been addressed and managed and corrected where possible. So we're talking about the kinds of patients where you would normally consider a transplant, for example. And to get on the transplant program here is quite difficult. You have to have all of these things in place before you're even considered. And I think the entry criteria for these kinds of trials where we're considering new sorts of therapies, whether it's stem cells or gene therapy, the patients will have to have received, a high level of contemporary care before they're eligible for these sorts of experimental therapies.
I wasn't throwing you under the bus, Eddy. I just realised I was talking to you.
And I just wanted to add the question that Katrina asked about how far we are from clinical trials. And I would just echo those comments that James made, that it's really hard to predict because the way these things evolve, there's a lot of ebb and flow. And, all it takes is one bad trial that can slow the field down for about 10 years. For gene therapy, there are a lot of exciting developments at the moment with the technology we're using. There's been a proliferation in clinical trials, and that's evidenced by publications in the New England Journal of Medicine, the peak medical journal for all of us. On this campus, there's been a lot of exciting development in terms of gene and cell therapy. There's been a lot of investment from the New South Wales government and from other sources to really see Westmead establish itself as a peak centre for gene and cell therapy. So I think Australia-wide, we're positioned in a very strong place at the moment in terms of infrastructure, intelligence, support, and the types of collaborations that James has been referring to.
I might just add one more thing about the lifestyle factor question. Westmead Cardiology, we have a really strong presence, not just at the basic science level, but in preventative cardiology. And one of our colleagues, Clara Chow, is doing great work. So across this campus, our research spreads that gamut from bench to bedside, right through to clinical trials, preventative treatments, app technologies, and we're proud of that.
So Eddy, practically, this is real frontier stuff: cell therapy, gene therapy, defang viruses, if you like, being used for gene therapy. You alluded to some of the resources that are needed in producing enough gene therapy vectors for treatment in the future. What are we going to need? And perhaps you might like to say something about the need for gene therapy vectors and perhaps a facility in Australia to enhance this because of problems around the world.
Yes, so I mean, essentially what we need is a facility that can produce large-scale clinical-grade vector, and at the moment that kind of facility only exists in fairly rudimentary forms across Australia. There is one next door, but that's got a very limited capacity. Probably doesn't have a capacity to run more than one small clinical trial at the moment. In North America, there are more centres that are established to provide clinical-grade vectors. But the waiting time, if you want to initiate a clinical trial, the waiting time to have your vector produced by one of these facilities exceeds 12 months, and you just get queued up with your order for these vectors. So I think a big thing that we need here, if we're taking gene therapy and cell therapy seriously, is the capacity to produce clinical-grade vectors or cells that can be produced in sufficient quantities to treat the kinds of patients we're talking about here. And that takes a big investment beyond the centre's budget. That takes government level investment with some input from industry as well where that's possible. Do you want to add to that?
I'll just add that for cell therapy, you need the right facility to produce treatments in a safe way. Now, for cell therapy, there have been less scrupulous practitioners who have used products that aren't really even stem cells. They've sold them as stem cells. They've created them in not that safe an environment. And they've actually been problems and in some cases deaths. So we need safe rooms, and here at Westmead Institute there is such a room that's being generated, and we're quite lucky to have that.
The amphibian that you showed earlier, there are other animals that are able to regenerate parts of their body. Why have you chosen that particular animal?
So we don't work with axolotls. I just thought they were kind of cute. but no, in all seriousness, zebrafish are other ones that people use because of their turnover. We don't work with them, actually. We work with more preclinical models that are closer to try and solve the hurdles to get into the clinic. So we don't actually work with it. I just used it as an example. Well, I don't know why people worked out that they could, maybe it was because of their limbs. They worked out that they could regenerate their limbs first. And then someone, actually a researcher from Melbourne, a good friend of ours, Enzo Perello, was instrumental in working out that mammals could actually do the same thing and regenerate part of their hearts. But we don't work with axolotls, actually.
Where do you get the cells from?
So, there are a number of different stem cell populations. We now have the technology, and Zoe mentioned this, where we can actually take your cells, either from your skin or even from your bladder, but via a urine sample. Then we can reprogram those cells to become stem cells and able to produce. So that technology exists, and we don't, here in the Institute, we don't do that reprogramming process, but other groups have done that, and you bring in the cells and use them for our experiments, different types of cells. Now that's only one type of stem cell population, there are others, but I think that gives a really nice example of how far we've come, what we can do with that technology.
So sorry, would the potential be that down the track, if I had a heart attack, you could take my cells and grow new cells?
That's possible. Researchers, that was floated as an idea. We're now thinking that, currently now, that will be prohibitive. The time that it takes to create your own cells, particularly for something like a heart attack, is going to be prohibitive. Currently, the cost is prohibitive to make your own cells, but in the future, who knows? You could have yours banked, the technology could become cheap enough so that that's possible. But so the concept's there, but right now, that's probably not feasible.
Can I speak to that? So there's been an idea that's been floated. There's a lot of labs around the world and companies now generating pluripotent stem cell lines from healthy individuals and from people with specific diseases. And I know that one of the possibilities that's been mentioned is pluripotent stem cell line banking. And potentially in the future, people who need cell therapy could be matched to a line that they'll have the least reaction to. It's kind of similar to the way that you get matched to an organ donor. So that's one potential way of getting around that issue of the time it takes to generate these stem cells from your own cells. So I think that's got a lot of possibility.
Other question, Usman?
Just be very futuristic. If we have all the resources in the world and all the scientific equipment, can we regrow a heart in the lab? And if those people who are inherited with heart disease, do you think that will there be a possibility one day they can actually ask the researchers or some companies to regrow the heart in the lab, and when they get the heart disease, then they can implant that heart? So just a bit of a question. Is there a possibility that we can regrow the heart?
I'll give you a short answer and then James can elaborate. So if you had a genetic abnormality, the technology exists, and again this is science fiction underpinned by a few facts. If we could take your cell, we could genetically correct the genetic abnormality you have using gene editing technologies and gene therapy technologies, and then we could hand that cell to James and he could grow up those cells into cardiac muscle and regenerate the heart that way. So that way you'd have a heart that was genetically corrected.
And that shows how far we've come in gene therapy. I never say never. I think that's possible. What you're talking about, creating a whole heart from the bench, that's pretty complex, because not only is it a number of different cell types, it's the spatial organisation, it's the electricity, it's the plumbing. So that it's a pretty complex task, but never say never. It's possible.
Should we? All right. So if there's no more questions, all good? All right, excellent. No, so I'm just going to invite Katrina Dowling now from the Westmead Institute for Medical Research Foundation to give us a few words.
Great, thank you. I'll be brief. I know that it's been quite a long session, but hello everyone. It's fabulous to see so many people here at WIMR on a Tuesday evening, so thank you very much for joining us. Welcome again to Meet the Researcher. It's now the third in our series of bi-monthly sessions. For those of you who I haven't met, my name is Katrina Dowling and I lead the newly formed WIMR Foundation here at WIMR. So I've come on board to establish the WIMR Foundation as a strong philanthropic foundation, supporting and facilitating the research conducted at WIMR now and on a sustainable basis into the future. So I'd like to say a big thank you to all of you for being here this evening, for dedicating your time and taking an interest in the work of our team of exceptionally bright, talented and very, very dedicated researchers. We hope you've been inspired and that you've learnt something new tonight. I know that I have, about the research conducted at WIMR and the capacity of researchers here to positively shift global health outcomes. James and Eddy, our chief electrician and plumber, lead one of the 11 research centres that collectively form the multidisciplinary research institute that is WIMR. And we investigate some of the most pressing health issues facing Australians and people throughout the world, with our vision being to liberate people from the burden of disease and serious illness. And this includes obviously people living with serious conditions, but also family and friends. So we exist to find better treatment, prevention and cures. So a big thanks to Associate Professor James Chong and Associate Professor Eddy Kizana and your team for such an informative session this evening. As the team have said, the heart is an essential organ and one that we can't live without. And with heart disease being the single leading killer of Australians, it's a really important topic for us all to be focused on and for us all to be doing something about to address. As James and Eddy have said, the symptoms of heart disease and heart attack are often vague, and they're not always what people would consider the typical symptoms. So there's many Australians who are walking the streets completely unaware that there's something going on with their heart, particularly the electrical issues that Eddy was talking about. And they don't know it until something significant happens. So education and awareness changes everything, as does medical research. So to achieve all that we can in terms of positively shifting global health outcomes, continued financial support for research is absolutely essential. All contributions make a difference, and there are many ways to get involved. Donating on a regular basis can help to provide a consistent flow of funds to ensure the continuity of research projects. Major funding, corporate partnerships, community support and workplace giving are all ways to get involved. Some people also choose to support our work by leaving a gift in their will. So there are many ways that you can get behind the work and support the team. Some of the WIMR Foundation team are here tonight, and I'm looking across to Hilary and to Icky. Please put up your hand, guys. We would welcome the opportunity to speak with you at any time if you are interested in supporting. And we've also got donation forms here tonight if you're interested in leaving a donation this evening. So in closing, thank you again to everyone here tonight, and particularly to Graham, who's made the special trip up from Melbourne. It's much appreciated. We appreciate you making the time in your busy schedules to share the evening with us and to learn more about the work that the team does here and how you can make a difference. So if you've enjoyed tonight, please tell others, and of course we'd encourage you to attend these sessions on a regular basis. I wish you all a lovely evening and a safe trip home, and we hope to see you again in the near future. Thank you. Thank you.